Thanks very much, Bruce and the organisers, for the opportunity to present again today. Thanks to the Minister for that very motivating opening address. And it's actually nice to be back in Perth. I was here at this meeting 5 years ago and learned a lot, and I'm learning a lot again this time around. And I'm going to tell you about some technology which has already come up at the meeting, and I think it's probably on many people's radar. And over the last 5 years, there's been actually some quite decent progress in this field. So I'm going to take you through that. It is technology with enormous potential, and I hope to put that in the context of how we might be able to use it in the future for cat biocontrol. So what I'm going to tell you about today is genetic biocontrol, or specifically so-called gene drives, what they are, how they work. I won't get into too much technical detail today. I don't think it's necessary, but happy to answer any questions on the technical details later. I'll take you through what species the gene drives have been developed in. Mostly it's driven by insects, insect control, but also it's now going over into mammals. And then finally, I guess the bit that you're most interested in, could gene drives be developed in cats? What do we know about that? How can we test that, at least using modelling? And what are the challenges and barriers to development? So I know this is a pretty diverse group of colleagues, individuals, so I thought I would just get us all on the same page, just genetics 101 for everyone, I suppose. And tell you a little bit, obviously, we all know about DNA, it lives in every cell, it's the blueprint of life. Cats have around 2.4 billion base pairs of DNA, and they're arranged onto chromosomes, which you can see we have pairs of chromosomes, shown here on the left-hand side. Now, everyone seems to have an image of a cat in their talk, and here's my image of a cat. It's not an experiment we've done, but this is a transgenic glow-in-the-dark green cat. It was generated by some researchers around 10 years ago. The point of this is twofold. One is to say that you can modify the cat genome. So when I talk about transgenesis or transgenes, what I'm talking about is modifying, effectively hardwiring a new piece of DNA into the genome of the DNA of the animal, and it usually does something interesting. In this case, it wasn't just about making the cat glow green in the dark, interesting as that is, it was used for a medical research question. The other point I'd make about this is that although we can modify the cat genome, it's actually doing a literature search on this. There's not many examples of this technology, so I don't think we can sort of underestimate the barriers to developing gene drives in cats. It involves transgenesis, and that's not very well established in cats, is my point. The last thing I'd say on this slide is that gene drives that I'll tell you about are a form of transgenesis. They're a transgene, so it's a modification of the cat genome. And essentially it breaks the rules of normal inheritance. So on the left-hand side, you can see here, you see I've got TG here. That indicates we've got a transgene or a bit of DNA that we've put into that particular chromosome. Normally that would only be passed on to 50% of the offspring because there's two versions of that chromosome, one with the extra DNA and one without it. What gene drives do essentially is they break that inheritance rule so that it always gets passed on to the next generation. Okay, so that's really depicted again on this slide, and what it shows you is the potential of a gene drive to either modify or suppress a population. So what I mean by that is, as I said, a normal gene will only be passed on 50% of the time. Eventually, these red ones will eventually be lost because there's usually a fitness cost associated with that. But a gene drive has a huge potential because you can modify the entire population because it always gets transmitted to the next generation. And it's this ability of the gene drive to either modify or suppress a population that really has people excited about the potential for landscape control of a particular feral pest. They work in a number of different ways. Again, I don't want to get into too much technical detail. We heard a little bit about that yesterday, but essentially there's two kinds of strategies that people are employing with this technology. One is to modify the populations in such a way that it can effectively spread a female infertility susceptibility modification so that you get to the point the population becomes saturated, those individuals then start to mate with each other, and that results in infertile females. That's quite an effective strategy. The second one is a male biasing strategy. Again, you might have heard of this. The idea here is to make the entire population male, which will cause a crash due to lack of fertile females. Now, why do we want to develop gene drives? Well, it's pretty obvious, I think, in the case of pest suppression, feral cat suppression. What's driving this technology, though, really is insects and malaria. So malaria causes about 500,000 deaths per year. So the idea here is that we can either modify the mosquito so it can't spread malaria, or get rid of that particular mosquito species from the population, from a local area, that will cause malaria to drop and therefore save lives. So that's driving a lot of the work in this space. Our particular work is on feral mice, and you can see examples of that here. We obviously have mouse plagues in Australia. They're pretty devastating, and they are something that we're aiming to address using this technology. Okay, so I could talk for hours about CRISPR. I'm not going to talk for hours about CRISPR, you'll be pleased to know. It's a fantastic technology. It's been around for around 10 years. A lot of the ideas I'm telling you about today have been around for around 20 years or so. CRISPR has enabled us to act on those ideas and to generate the organisms to test this kind of technology. And in terms of the insect world, let's grab a glass. In terms of insects, we are really in quite a decent position in terms of technology development. But it's got to the point now where they're doing cage trials of gene-drive technology that's really suppressing the population of large numbers of mosquitoes to the point where they're becoming eradicated in just a short number of generations. So there's real potential here, I think, to use this technology for malaria control. But what we do know from the last few years, from the work from my group's been doing, and also other groups, is that these so-called homing drives, which is the technology that's used here, is not really working very efficiently in mammals. So the technology that we've been working up just recently in my group is called the T haplotype. So the idea here is that we can spread a version of this natural gene drive. So this is a technology that will only work in mice, so we can't translate this over to cats, unfortunately, but perhaps there will be cat versions of this, we just don't know at the moment. But the idea here is that we use this so-called T haplotype to spread a female infertility gene through a population. And the way this one works is that it affects sperm development such that the sperm that carry this natural drive win the race. They essentially nobble the sperm that don't contain this particular DNA element and results in their winning the race to fertilisation. Therefore, we get spread through a population. So we've really had this concept, and we're trying to understand, could this work in the context of a gene drive? Could we modify a population using this approach? We decided to engineer it using CRISPR to spread a female infertility mutation, but didn't really understand if it could work or not. And this led us to using a spatial modelling approach, which we developed with Ashikul Baran. So Ashikul's a fantastic modelling postdoc who works at the University of Adelaide. And she developed this model that essentially goes through a breeding cycle. It can be made specific to individual species. So we can use it to test cats, which I'll tell you about in a minute. Essentially, it's an individual-based, spatially explicit, stochastic model, which means that we trace individuals within the population and ask the question, can we use it when we add particular gene drive individuals to this modelling system, can we use it to eradicate the population? Essentially, there's a whole bunch of events that go through, which are the life cycle of the breeding cycle of the individuals. And as I said, they're traced within this modelling structure. And there are 200,000 in this particular model that we use. So to show you a little bit how this pans out, how it looks like. So what you're looking at is a hypothetical island. On the top left, there's a number of patches on the island, and they contain individuals. And the red individuals that you see here, they're the gene drive individuals that we've seeded onto this virtual island. And you can see that the gene drive spread because everything becomes red after a period of time. And the population is over here. You can see it declining across time. And the size of those dots indicates the number of individuals in each patch. So you can see eventually it declines. And hey presto, after a number of years, you get eradication. So I really like these experiments because the mouse experiments take, years and years to perform. We can do this modelling in a week or so. So it is a very powerful tool to help us understand the potential of the technology. So that's so far so good for mice, and we actually engineered this strategy. We published it late last year. I don't want to go into the details of that today, but this is really an example of the first proof of concept for a gene drive in a mammal, so we're often running in terms of the technology in mammals. So that's very exciting, but it also raises the question, what about other invasive species, and in particular cats? So we decided to use the model to essentially have a look at, can we use the same technology that I described earlier, the classic gene drives, either the male biasing drive or the homing drive that spreads the female infertility gene. Can we model those strategies using the same number of individuals that we've been using for the mice experiments? So 200,000 individuals. That's quite a large number of individuals. So when you think about the density of the individuals in the landscape, these invasive mammals, with mice, 200,000 is quite a small number, it's quite a small area. Whereas when we think about cats, because the densities are so much lower, that's quite a large area. So I'll come back to that point in a minute. But the number of individuals is the same across all of these modelling experiments, which I'll show you in a minute, all the way. So it's mice, rats, rabbits, cats, and foxes. That was the experiment. We also had to, I guess, make our best estimate of what are the critical life history parameters that we needed to plug into the model to get the best representative result. I fully acknowledge it's a model, it's not perfect, but we're trying to get the best, I guess, estimate of what could happen in the environment if we introduce these gene drive individuals. So as it turns out, there's a whole bunch of data that we've taken from the literature. If you have any issues with this, I'm very happy to give you Ashgal's email address for you so you can contact it. No, but seriously, we've done our best to really make an estimate of what is the most realistic as possible. And again, to come back to this point here, this is the area. So this is about the size of Norfolk Island, and this is larger than Tasmania. So we've got the same number of individuals, but the size of the experiment is really quite different. Okay, so what did we find? So this is a cat simulation, and as I said, it's an area larger than Tasmania. It's 100,000 square metres, and we've used one of the classics. So this is the so-called X-shredder strategy, male biasing strategy. We've introduced the individuals, just 256 at the start of the experiment, the modelling experiment, and you can see what happens is that we essentially create holes in that population as the gene drive spreads through, creates an excess of males, and you get population loss. So a couple of things to point out. One is that it's quite an effective strategy, but the time periods here are huge. So I talk about 200 years here. Now, you've got to take that estimate with, I think, it needs some context around it. And that's all summarised here on this slide. So the take-home message really is that this kind of technology is going to be a lot faster in smaller animals, smaller mammals, invasive, particularly mice and rats. Rabbits are not so different in terms of the time frame, but the time frames are larger when it comes to these individuals that have greater survivorship, longer generation times, etc. So dispersal was another key factor we found as well. So if the smaller dispersal range limits the spread of the drive, which creates longer times to eradication. Nevertheless, though, I think it's important to note that we have some interesting figures over here. So this is the time it takes to get 50% of the population reduced, and you can see those numbers come right down. Although they're still quite long time frames, you can think about now doing this experiment on much smaller populations and asking the question, could we get eradication in shorter time frames? Okay, so this is my last slide. This is really essentially where the field's at the moment. So genetic biocontrol is progressing pretty well in insects and mice. We have examples now that we can point to and say there's a prototype that we could take forward to the next stage. They've got enormous potential for control of invasive species as well as species that cause human disease. There's a whole lot of other areas that I haven't touched on today that are related to this technology that are very important. They include stakeholder engagement, obviously, regulation, because we're dealing with a GMO here, and there are still technical hurdles to their development. And one of the key ones is to ensure that you're not affecting the native range of a particular species that you're looking to target. In terms of cats, clearly the time frames are longer than we get for the smaller mammals, and there are quite significant technical challenges associated with working with cats. We just don't have a really good handle on the techniques to make the transgenic cats. That shouldn't be underestimated. But that being said, and clearly they're a domesticated non-model companion animal, which does create other issues as well. But I'll leave you with, I guess, two points that I think that are quite encouraging. There was a recent survey by CSIRO. I'd encourage you to have a look at it. It was by Aditi Mankat, looking at public views towards gene-drive technology in cats. And the take-home message really is that around 2/3 or more were quite keen on the technology. They felt that it was something that should be considered, which I think is a good sign in terms of the landscape going forward. And the other thing is we need much better modelling data than we have already. We've done the best we can, but I think additional field data, including population genetics, I know CSIRO are gathering data on that, will help us to inform the modelling to make even better estimates of the potential for this technology going forward. So with that, I'll thank you for your attention, and just lastly thank the members of my group who've done the work. I just want to point out two people. I don't have a photo of Ashwell, unfortunately, who's done all the modelling, but the molecular work, which I didn't really get much chance to talk about today, has mainly been done by, there he is, Luke, a PhD student in my group. And lastly, just to thank collaborators and funding sources. Thanks for your attention.